Hello guys, I welcome you all to Biology Practicals. With me I have Pappu, and today you guys are going to have fun because we are going to do lots of experiment, starting with the first experiment that we have, and that is pollen germination. So let's get into it.
So children, today we are going to see the pollen germination, and in this experiment we are going to do only few basic steps that you need to know. It's a very easy experiment. So it goes like this. The first thing that you would require is vinca rosea. The flower looks like this. It's easy to observe, which is why we have taken that. And now we are going to use a needle, or you can also use a brush to shed the pollen grains onto the next thing, which is a glass slide. But it is preferred that you use a needle because since the pollen have the adhesive properties, it can go stick on the brush and it might be difficult for you. Okay, so now once we put the pollen grains onto the slide, we also need to provide the nutrient medium. Only then it is going to germinate. And what is that nutrient medium? It is going to be the sucrose solution. Post that, we would put a cover slip and we would observe it under the microscope. Also children, please make sure you would obviously be using your microscopes, right? And it is recommended to use the microscope under the power of 10x. So you do not need to look for 45x, just 10x would be more than enough. And I hope you have seen the microscope. It looks like this. So there is 10x power and there would be 45x. Go for the 10x one. And now we are going to see the experiment in real. Let's get into it.
So first you would need the vinca rosea flower. You can dissect it like this or using a needle. Then with the same needle we can shed off the pollen grains onto a very clean glass slide. And then with the help of a dropper, put 1 drop of sucrose solution. Post that, put the cover slip, and while putting the cover slip, make sure there are no air bubbles. And please wait for 10 to 15 minutes so that the pollen germination process can take place with the help of this nutrient medium. After that, you can view it under the microscope in 10x power and it will look something like this.
Okay chams, so I hope you guys had fun doing that experiment. Well, now let's check out what could you actually see under the microscope. So yes, definitely so many pollen grains would be there, and some of them might have started the germination process, as in the process where they will be forming the pollen tube. So over here you can see tube-like structures being formed, right? So we say that these pollen grains which are there, they have started off the germination process. So this usually happens on the surface of the stigma. Once the pollen grain lands on the stigma, basically the stigma would be providing some nutrient medium for it such that it could grow the pollen tube. But as of now, we are not providing any stigma out there. We are providing a nutrient medium directly, and that is sucrose. So with the help of this nutrient medium, in the laboratory condition, it is going to start off the germination process. That is all about this particular experiment. Now, obviously, in your school, there might be different steps that they might have shown you. Please talk to your teachers who have done the experiment and then only go ahead. Okay, so the general steps which are mentioned in the lab manual, I'll be talking about them also. So according to the lab manual, it says that take a drop of 10% sucrose nutrient solution on a clean cover slip. So they are taking on a cover slip and they are asking you to dust the mature pollen grains of the vinca rosea on this particular cover slip, and then to place the cover slip inverted onto what? A clean slide. Now that can be a little troublesome for all of us, which is why there was an easier way and we showed you that. Okay, and please make sure that you keep the pollen grains undisturbed for 10 minutes because obviously they have to take in these nutrients and go ahead with the process of pollen germination. I hope that is clear. Now, what will give you marks in this particular practical? You need to write some things up, some very important things, and I'm going to talk about that. But before that, I want to ask, haven't you subscribed to the channel yet? If at all not, then please subscribe to it right away for getting all the important videos. So the preparation of the slide will give you 3 marks. Okay, the experimental things that we showed you right now, if at all you do all of them correctly, you will get 3 marks. Now, you need to have 2 more marks because this whole experiment is totally out of 5 marks. So where will you get your 2 marks from? Listen to me. If at all you make this table for calculation properly, you will get 1 mark. What is this table about? So this table is about the total number of pollen as well as total number of pollen germinated. So suppose your slide looks like this. Okay, suppose your slide looks like this, and this is your area where you are checking the pollen grains, and you have multiple pollen grains out there here and there. What do you have to do? Divide them into 3 columns, 1, 2 and 3. Okay, now check for the total number of pollen grains in this column number 1, column number 2, column number 3, because over here you have to write 1, 2, 3 observation. So in the first one I see the total number of pollen grains which are there, they are 3 in number. In the second one I see 1, 2, 3, 4 pollen grains are there, so I write it down. Similarly in the third one I see 2 of them are there. Now that is capital N. That is what? Capital N. That they are also asking you total number of pollen grains germinated. So you have to check now in each column how many of them are germinating. Suppose over here 1 is germinating, over here 2 of them are germinating, over here again nothing is germinating. So what do you have to do? Check for each of the column and write accordingly. So in column number 1 only 1 is germinating, so I write 1. In column number 2 I see 2 of them are germinating, so I write 2. In column number 3 none of them are germinating, so I write 0. Now what do you have to do? You have to do a total of this. So 3 plus 4 plus 2 is equals to what? 9. And over here 1 plus 2 is equals to what? 3. It would be equal to 3. You got the totality of it. If you make this table for calculation, you get 1 marks along with the content within it. Okay. Now, your next 1 marks depends on 2 things. One is the percentage of pollen germination. You have to calculate the percentage of pollen germination. Now, if at all you write this formula properly and you replace the terms over here within this particular formula, you are going to get your half marks. So, let me replace. So, small n is basically, so I have to write n is equals to 3, capital N is equals to what? 9. Now, you replace it. So, small n is equals to 3, 3 into 100 divided by capital N. What is capital N? 9. And you do the calculation. So replacement of all these terms is important, gives you half marks. Along with the formula also you have to write this down. Next is calculation of the pollen germination. So you do the calculation and that will give you what? Another half marks. Got it? So this is how it usually goes. So half plus half, 1. You already got 1 from here. So 2 marks done, and 2 plus 3 is equals to 5. This is how you are going to get your 5 marks in this particular experiment. I hope it's clear. Okay, do not worry if at all you do not see pollen germination. You can just write 0, 0, 0 out there. And also if you have any doubts, talk to your teacher also about it once before you enter. Okay, but do not panic. Works? So this is how it goes, my dear children. Also, one very important thing, I have already shown you the name of the flower in the video. It is Vinca rosae, and this is what you will get to see, the pollen germination happening. Also, one very important thing, we have as of now done the first experiment over here. So out of 4 you would be asked to do any one experiment, and you cannot choose that. It is totally upon lottery system. So we have done the first experiment on pollen germination, and now if at all you want to see the next experiment, do not forget to check our channel and do not forget to hit the subscribe button. Also, if at all you are a KCET aspirant, my dear children, I request you to go through ABHYAS KCET test series. It is definitely going to help you out. There are 35 KCET mock tests and also chapter-wise KCET PYQs, and all of it under proper exam-like condition. And all of it is for rupees 1499. The link will be there in the description section. Do check it out as well. Okay, so that's all for today, and wishing you all the best. And if you have any doubts, put it down in the comment section right below.